On air.
Okay, today we're going to learn about translation in RNA protein synthesis. Of course, this starts out with DNA. You know, your double helixes has to split, and when it's split, it makes an mRNA cell, which is messenger RNA. And I'm moving a little bit fast, so you guys will kind of be lost. But I'm going to show you a video that's going to show you exactly what I'm talking about. But what do you guys know about protein synthesis? Do you know any background of it, even a little bit? Nothing at all? Okay, so with this process, you have DNA. DNA is split by helicase. Can you say helicase?
Helicase.
Helicase. Okay, so it's split by helicase. Once it's split, you know, a big process goes on, and that makes your messenger RNA. Messenger RNA is then sent to form your tRNA. It's going to merge with your tRNA, and I'm going to show you how this basically that process, and I'll break it down to you. Now, this process has 3 separate steps. Those steps are initiation, elongation, and termination. So, what were the 3 steps?
Initiation, elongation, and
termination. And termination. Good. Okay, with these 3 steps, of course, I'm going to break down everything that's going on. These 3 steps are basically that diagram that I drew down there. All right, now, to break this down just a little bit more before I actually explain it so you'll know what's going on, these boxes here are amino acids. Those amino acids contain adenine, thymine, cytosine, guanine, and sometimes uracil. With these, they bond to specific types of amino acids. So the adenine, well, of course you know what these letters are. So what would this mean?
Adenine, uracil.
Thymine with adenine, cytosine, guanine, and T is replaced. Thymine is replaced by uracil.
Right. So basically, those equal signs are the adenine bonding to uracil, the thymine bonding with adenine, and so on. So, with that being said, let's try a sequence. Okay. So I gave you 3 amino acids. Now, I want you guys to translate these based off what I just showed you down there. So the A would be
U.
Would be what?
U.
The U would be
A.
G.
C.
G.
T.
U. A. G.
Good. All right, now that you guys have that part, I'm going to actually explain the actual process. So, here's your DNA. Your DNA is broken by helicase. Once it's split, in order for it to stay split, you have proteins that keep it apart in order for it to not spin back again. So this is your mRNA, which is basically from where?
One side of the helicase.
One side of the DNA, and the helicase broke the
one side of the helix, I'm sorry.
Right, correct. Okay, so this process is called transcription. All right, once your mRNA moves up to, this is a ribosome cell, this circular part. That mRNA goes into this cell. You have 2 parts of it. You have a small subunit and you have a large subunit. And once this mRNA goes, it's basically somewhat of the same thing that I showed you guys down there. But once it goes in, these amino acids starts to translate. When it starts to translate, you have these T cells, which is tRNA. Okay, so multiple tRNA cells have amino acids at the bottom that binds to these, which is basically what we did down there. Do y'all follow what's going on? Okay, so Once this part starts here, this part is the initiation process here. Once the TNA cells come in, each tRNA cells come in. Each one of them has a portion of the tRNA cell that contains a specific component that is going to begin to bind together when each one come in. So say you have this one tRNA cell to come in to merge with AUG, so that means that this amino acid has to be what? If it's going to bind with AUG, what
Has
are
to
the...
be U, A, C.
Good. So once that binds together, this tRNA cell is going to get ready to leave the cell, and another tRNA cell containing another component is going to come in and bind to that one, and it's going to change this to bind with that, and it'll be what?
CCU.
Excuse me?
CCU.
Okay. CCU. Once this tRNA cell come in and join here, and it contains the CCU, this cell is going to break off this tRNA cell. This tRNA cell is going to leave. This one's going to bind, and then this component is going to bind with the A site of that component. So therefore, these two are going to end up merging together. They're going to be connected. That part is the elongation process. This is going to, the tRNA cells are going to continue to come in. This ribosome is going to continue to move down the sequence until it reaches the end of the sequence. Once it reaches the end, there's going to be a cell called stop codon. It's going to be a stop codon, and a release factor is going to come into the ribosome and make this process stop, and that's called termination. So, what part is where it begins? Initiation. Once it starts to produce this new protein, when the tRNA cells, those A sites merge together, what is that called?
Elongation.
Elongation. And then once it reaches the end and the release factor comes in, what is that called? Termination. Once it reaches the termination process, that's when the small unit and the large subunit of the ribosome cell is going to disassociate and the process is complete. And that's called translation. So, what are the two parts of RNA that has to happen?
Split.
Okay, split. Transcription. Translation, but what happens with translation? What's before elongation? Initiation. Elongation. Termination, and then?
Translation.
Translation is what's going on in the ribosome. Once it reaches the end and reaches your stop codon is termination, and then once it reaches termination, what happens to the ribosome cell?
They disassociate.
Disassociate. Y'all did really good. That wasn't that bad, was it? Go me. Okay, so now that I've showed you guys that, I'm gonna let you see it in the actual video that I made, and I want you guys to call out what's going on. Hopefully you can't hear it really, really good, because I want you to tell me what's going on. It's basically what we just went over.
I'm videotaping a video.
Okay, thank you. Okay. How do you make it pause?
Pause? You probably have to wiggle the mouse and get a pause button.
Okay. So the ribosome that you just seen